Now here we are concerned with the nitrogen bacterial biofertilizer, that is Rhizobium. Then as far as the Rhizobium is concerned, Rhizobium is a bacterial form of a biofertilizer which is responsible for nitrogen fixation in the particular type of plant. That means these bacteria, which are aerobic in nature, gram negative in nature, it is growing inside the roots of leguminous plant. That means members of family Leguminaceae, all the members of family Leguminaceae contains a root nodules, or develops a root nodules on the root, and these root nodules having a number of this Rhizobium bacteria. That means Rhizobium bacteria infects the roots of leguminous plant, and this after infection, this Rhizobium simply develops a root nodules, or the root nodules are formed on the plant, and this Rhizobium survive in the root nodules. And then after, this Rhizobium is responsible for fixation of free nitrogen, that is N2, into a available form to plant, that is NO3, or in the form of nitrate. That means Rhizobium is responsible for fixation of atmospheric nitrogen and conversion for the plant. That means it is utilizable form to the plant, and then absorbs it will be absorbed by the plant after it is formed into the NH3, that is in the form of nitrate. Then as far as this Rhizobium is concerned, Rhizobiums infects a different plants, different leguminous plant. For example, there is a specificity. For example, Rhizobium physioli responsible for infection to the bean plants. Then Rhizobium leguminosarum responsible for infection on the pea plants. Rhizobium japonicum is responsible for infection on the soybean. That means these are some specification of the Rhizobium species on the plants. Some Rhizobium species are responsible for infection on the groundnut plant, pea or sisal. That means different species are there and different specifications are there, and this Rhizobium simply infects the member of family Leguminaceae. Then this, after infection, the N2 fixation starts. The root nodules are developed on the plant, and then N2 fixation starts. N2 fixation starts when root nodules are developed on the roots of the leguminous plant. Then there is one pigment which is developed in the root nodule, that is called as a leg hemoglobin. These leg hemoglobin pigments are simply responsible for fixation of nitrogen, or this leg hemoglobin absorbs the molecular oxygen and it makes available to the Rhizobium. Because Rhizobium is a aerobic bacteria, it can only survive in the presence of oxygen. That means oxygen is available by the leg hemoglobin to this Rhizobium bacteria, and then this Rhizobium multiplies in the root nodules. Then due to the hemoglobin, leg hemoglobin, this oxygen is available, and due to that the nitrogenase enzyme is developed by the bacteria, and this nitrogenase is developed by one gene present in the bacteria, that is called as NIPE gene. That means NIPE gene is responsible for synthesis of nitrogenase enzyme in the bacteria, and this nitrogenase enzyme is synthesized because nitrogenase enzyme is cannot able to absorb the molecular nitrogen, and the nitrogen whatever may be developed by the leg hemoglobin or modified by the leg hemoglobin is simply absorbed by this bacteria, and then nitrogenase enzyme is synthesized, and this nitrogenase enzyme simply responsible for modification of this nitrogen, free nitrogen, into a NO3 and NH3, that is nitrate form. Plant can use only the nitrate form, and due to that this fixation takes place, and after that this plant utilize this nitrogen, and due to that the soil fertility increases, the growth of that particular leguminous plant increases. That means near about 20 to 40% more growth takes place due to the available nitrogen by this bacteria in the root nodules. That means root bacteria is responsible for more growth of this leguminous plant, due to the fixation of nitrogen in the root nodules. Then as far as this bacteria is concerned, this bacteria fixes the nitrogen, and after the cultivation of this plant, crop plants or leguminous plants, the remaining nitrogen is fixed in the soil. And this is the actually role of rhizobium in the plant, leguminous plant. But as far as the cultivation of this rhizobium, artificial cultivation of this, or artificial production of this mass cultivation of this rhizobium takes place with the help of following methods. For example, if the bacterial culture, pure bacterial culture is cultured on the yeast extract medium, that is mannitol broth medium, the bacteria multiplies near about in 9 to 10 days. That means maximum multiplication of bacteria on this medium takes place in the 10 days, and large number of bacteria is developed in the yeast extract mannitol broth medium. Then after this number of bacteria, or this pure culture of this bacteria on the yeast extract medium, simply this extract is mixed with the carriers, some carrier substances like lignite, charcoal, sometimes farm manure or compost is also used for as a carrier. Sometimes charcoal is used as a carrier, sometimes lignite coal is also used as a carrier, sometimes also there is a cocoa peat also sometimes used as a manure, used as a carrier, and the powder is simply packed in the plastic or polythene bags, and then it is marketed or it is supplied to the farmers. Then this is the mass cultivation of this bacteria. If this mass cultivation or this biofertilizer is used by the farmers in the field, at that time if it is used for seed dressing, at that time there are some benefits of this rhizobium bacteria. For example, if this material or this fertilizer, biofertilizer is used for, or it is used for seed dressing, at that time the seed germination is more and seed germination is also fast. That means seed germination is more, seed germination rate or growth of that seedling is more or fast due to the dressing of this biofertilizer, that is rhizobium containing biofertilizer used for the seed dressing. Then second, this biofertilizer receives more nitrogen and develops a more growth hormone. That means nitrogen percentage is increased, growth hormones are also developed in the seedlings with the help of this seed dressing to the seeds. This treatment, seed treatment, simply responsible for increasing the soil fertility at the same time, and also responsible for increase in the hormonal percentage in the soil. Then this particular treatment, biofertilizer treatment to the seeds, also responsible for increase the disease resistance in that plants which are cultivated in the field, and also it is responsible for more yield. That means these are some benefits. For example, the seed dressing with rhizobium biofertilizer, seed germination percentage is more, seedling growth is more, in the seedlings nitrogen percentage is more, growth hormones are developed. Also in the soil, growth hormones are more and nitrogen percentage is also more. That means simply due to that seed treatment, soil fertility is increased, crops are having more growth, then crops are having more disease resistance, and ultimately all that particular benefits are responsible for more growth in the plant. That means this is about the rhizobium. Rhizobium is a biofertilizer. Why? Because it is simply present in the leguminous plant, and these root nodules are having more bacteria, or if suppose artificially cultivated on the extract, pure cultures are developed, at that time also it is useful, and it is ultimately responsible for more yield in the crops by the farmers.